One of our curiosity questions that somebody asked was, well, bats hear differently than us. Well, do they see differently as well? And are bats really as blind as a bat? Well, to understand this, we need to understand a little bit about how we see. You've got your eyeball here, and light comes in, and it projects back on here on your retina, and this is where we have these specialized nerve cells that allow us to see. You've probably heard of rods and cones. These are little parts back here that are on the back of the eye that are sensitive to the light, and they send signals into your brain. Well, up close, if you were to slice through an eye and take a look at things, this is a microscopic view of them, and what you notice is that they're called rods and cones because that's actually the shape of them. They have sort of a cone-like shape here where the sensitivity is on the outer part of that cone for the cones, and the rods are there. And one of the things you notice is that there are a lot more of those rods than there are cones, and that is indeed true. And there's some differences between them. So in our humans here, the rods, those are help actually on everybody. They help the low light and black and white vision. And there are a lot of those in us. And in fact, even in humans, it's 90% rods in our eyes. It's about 150,000 rods per millimeter squared is what I read. But in addition to those rods, we have our cones, and the cones are divided up into three parts. Some of them are sensitive to short wavelength light, like blue colors. Some of them are more sensitive to the medium wavelength light, green, and long wavelength, red. And so we have three different sensors, which allows us to put light together in different combinations, and so we can actually see different colors. If your short S cone sends a signal, that means, and the long L cone does not send a signal at the same time, that means it's probably a bluish color light. If you've got a green and a red, the M cone and the L cone send signal at the same time, that could very well be a yellow light that's triggering both of those in about equal quantities, and so your brain says, yellow, and that's just how you see color. Well, bats, they have a lot of rods. About 98 to 99% of their eyes consist of rods, which makes sense because they use a lot of low light vision. And even still, they have hundreds of thousands of them, way denser in terms of the rods per millimeter than human eyes do, up to 800,000 rods per millimeter. So good night vision there. But they also have cones, and most bats have two types of cones. And one of the consequences of that is that they're going to see less variety of color. They're not going to be able to put together as many combinations of colors, so they're not going to be able to see quite as much in the way of color. But the other interesting thing is, particularly with nectar-eating bats, the S cone, which is the short wavelength, has pushed a little bit further over to the even shorter. The ultraviolet light is visible. So nectar-eating bats can actually see in UV, a lot like bees can, and that makes sense because I guess our flowers have evolved to provide beautiful patterns and colors to attract pollinators to them, and bats have picked up on that and have been able to see and find those beautiful colored flowers a little bit more easily, just like bees can. And I did read that some bats do lack the S cones, which mean that they would have just one type of cone and one rod, and they really wouldn't have very much in the way of color vision. And so can bats see as well as humans? Well, they can't see as many varieties of color, but they're really good at seeing in black and white. Now, the other thing that might be exciting is vampire bats. You might have heard that vampire bats can sense heat. And it turns out they can, but not with their eyes. If you look at the vampire's cute little nose leaf here, it has a ribbon of sensors, of nerve sensors there that go along that, and that seems to be basically consists of similar nerves that we have all around our skin to detect hot objects. If you put your hand near something hot, you can feel it even before you touch it. That is good. The bats have that same thing, vampire bats have that same thing in their nose leaf, but it's even more sensitive. It's tuned to lower temperatures. And of course, why would you want to have that if you were a vampire bat? Well, the vampire bats are looking for the warm blood flowing through your veins and trying to find where that's localized so they can bite the most efficient place to bite, and so they can sense that and kind of go in there. And if you've ever had blood taken or an IV put into your arm, you know that one of the challenges for those folks is trying to find your veins. Well, if they had this beautiful nose leaf temperature sensor, that would help them figure out where to go a little bit better. Another curiosity question that somebody had was about the social calls of bats, and in particular, can bats, learn anything from each other? Echolocation is the main way that the bats send out signals that we can record, but they do send out other signals as well. We're going to talk about social calls a little bit later, and so instead I actually wanted to show you the results of a cool experiment answering this related question: do echolocation calls include any other information in them? And so this was an experiment with big brown bats, and what they did was they took big brown bats and put them in a little cage, a mesh cage, and let them hang out there for a bit. And while that bat is in there, it's going to be checking out its surroundings, and it does that with echolocation. So it's going to be calling, and so they'd observe how often are they echolocating in that cage. That's the baseline. Then they're going to play back the call of another bat on a speaker, either a male bat or a female bat, and they're going to observe how often that bat echolocates after they've heard this thing. So there was a baseline, there was this strange other bat sound, and we want to know, what does the bat do in response to that? And look at how much their activity changed. And so they also, just for completeness, they did that with a male bat the first time, let's say. They're going to do it for the female bat sound the second time, so that they can really kind of see if there's a difference. Can bats tell the difference between the male call and the female call? Do they react differently to those? And they repeated that 15 different bats with different combinations of sounds so that they could really hone in on this question: can bats tell whether another bat is male or female? And I guess one other thing I should say before we do this is, it's if you're holding a bat, it's pretty easy to tell whether it's male or female. They have genitalia that you can see there, and so you can then go ahead and figure out, okay, this is a female bat, and then you can record its echolocation calls, and then you know for sure that you're playing a male call or a female call there. So that's something that I forgot to mention, but is an important part of this, is how do you know whether you're playing a male or female call? Can you tell the difference? Well, I can't tell their difference in their calls, but can the bats? Let's take a look at the results. So what this is showing here is the rate at which the bats were calling relative to their baseline. So the before versus after. And if the before and after are the same, you show up along this little yellow line of 0. And when the playback was of a male bat call, sometimes the bats called more often, sometimes they called less often, but on average, they pretty much said, I'm sticking with the same rate that I was calling before. And so not any difference. They don't seem to react much to that male call. Interestingly, when they heard a female call, they actually would call less often afterwards. And there's, the authors of the study say, okay, clearly what that means is that bats can tell the sex of a bat from its echolocation call. They can hear that difference, and they say, given the lack of useful visual information, because you can't see very well at night, you certainly can't see the genitalia of the other bat, the ability to determine the sex of an unknown bat by its sonar calls might be especially useful. And I think that makes sense. They also go on to speculate, why might they call less often when a female is around? And one of the things that that we do know is that sometimes bats will be a little bit territorial, and they'll try and get rid of their friends by trying to jam sonar. Basically, if there's another bat in the area that they don't want around, they can make a lot of calls, and then it makes it really hard for that other bat to hunt, because remember, they all have about the same frequency call. And so if you want to get rid of this other bat from your territory, you can make it really miserable for them to hunt. Well, the opposite could also be true, where if you've got a female in your area that's hunting along and you want to be courteous to it, you don't want to chase it out of your territory. Maybe you want to call a little bit less often, so you're interfering with it a little bit less often. And so that was one of the things that they speculated might be the case, but clearly a fun area for more research for anybody that's interested in going into bat acoustic research. All right, let's dig in a little bit more. The next thing that we are having our folks do is we need to understand a little bit about how we can use our computer to build these spectrograms. And last time we were able to load in data and plot the raw data. But this time we want to get down to how do you figure out what frequency something is. And so let's imagine that we have the plot of a waveform of here, where time is on the x-axis and the intensity is on the y-axis here. And so imagine you have a 40 kilohertz wave. This is not a 40 kilohertz wave unless we have a really long thing, because 40 kilohertz wave is going to have lots and lots of cycles per second. How many cycles per second is it going to have? 40,000, of course. That's what 40 kilohertz means, is 40,000 cycles per second. So a related question is, how many wave crests in 1 second? Well, you know that a cycle consists of one complete crest and one complete trough going back to the beginning, and that means that there is one crest for every one cycle, which means if you have a 40 kilohertz wave, you've got 40,000 crests that should go by in 1 second. Now, we want to be able to do things like take a look at a waveform and count the number of crests, and I don't want to count 40,000 of them. So maybe if I took a shorter time sample, instead of a full second, what if I took 1 millisecond, 1,000th of a second? How many wave crests would I expect to see in this 40 kilohertz wave then? Well, that's 40,000 cycles per second divided by 1,000th of a, or divided by 1,000 pieces of a second, so 1 millisecond. That means that it's 40 wave crests in 1 millisecond. So now if I plot 1 millisecond worth of data, I can actually count the number of crests in there and get a pretty good idea of what the frequency is. So let's go ahead and give that a try. Well, here I am in R, and I have my script that I'm going to be using up here. My console is down here, and my plots are going to show up over here in the R studio. And I'm going to, first by making sure that I'm clear on where my directory, where all my files are stored is. So I'm going to hit enter with that. And then I'm going to define my Batwaves library that you can download from Canvas, and I've got it called batwaves.r, and there's the library file. And then we have one wave file, the first one that my wave exploration number 1, or wave1.wave. I'm going to define that. And what I can do then is I need to, this command here is going to activate the library or run all those commands so that it's going to train the R how to do bat things with my library file. And then this next one, now that I've trained it to do bat things, I'm going to first read in the audio file and store it in a variable named audio. And so if I type, if I go down to my console and I type audio, you can see that it's stored in this file here, and it has a sampling rate of 340,000 hertz. We'll talk about what that means a little bit. It's got 3,841 samples, and it's a mono signal as opposed to stereo, because there's only one channel of it, and it's a nice signal. So we're going to go ahead and then get the y values, or get the amplitude values out and store them into the variable y by doing this. This says take the left channel, since it's only mono, it's stored in the left channel, and save that as a variable y so that we can create a simple plot of it. Plot y. Pretty cool. Unfortunately, this is all a bunch of circles, and we need to tell R to please make those into lines. And so we can ask it to do type equals L. And if you look at your cheat sheet in R, there's a whole bunch of instructions on how to make the way a graph looks different. So now if I hit that, you can see I've zoomed in quite a bit. Now, I can also then, or sorry, not zoomed in, I'm sorry. I've created the lines, but now I need to zoom in to see those lines so I can see the wave crests. And so what I can do is I can say the x limits, and I can tell it I want to look at, say, from 1,000 to 2,000. Let's zoom in on that section, and I can hit enter. And you can see I can start seeing my wave crests. Or I can zoom in even further from 1,000 to 1,200. Oh, interesting. You can see that the waves seem to be a little bit different than before. Maybe I should go a little bit lower than that. Let me go to, and I hit the up arrow, and I can rerun my command that I was on before. Let me look from 900 to 1,200. Interesting. So we said we were going to be counting wave crests here, and let's see, do we feel like the frequency is changing here? Well, if you look at these waves, they seem to have the same frequency. They repeat at the same interval. Some are just taller than others. In other words, they have a higher amplitude. So we've changed the amplitude but kept the frequency the same for this. So we should be able to just count the number of wave crests, except that there's a problem. We need to count the number of wave crests in 1 millisecond. What's stored on our horizontal axis here is called the index in this case. Because if I go back to just plotting this as individual points, what happens is that the way we record a digital signal of something is, in fact, let me zoom in here actually. Let me show you this like this. Let's get rid of the, let's zoom into that same section, but we'll zoom in here. Can you see these dots? They are indeed, if you connected them all in order, you would get the exact picture that we were looking at before. But a digital signal is actually, what is happening is you're recording the intensity of the signal at a whole bunch of different times, a bunch of different samples, and they happen at regular intervals. And so we're going to look a little bit at how that works here to talk about it. So the true signal goes up and down, consistently. There's energy at all moments of time, but we're only recording it at specific intervals. And so let's say that we have here a 2-second signal. We're no longer looking at our 40 kilohertz wave, and we have 2 seconds of signal, and we're going to be recording every so often here. This looks like it's every quarter of a second it records. And so the question is, looking at this signal, first of all, this signal, how many hertz is this signal here? That's what we're looking for. How many cycles per second does it have? And if you look, there's 1 second and there's 1 cycle. Oh my gosh, it's a perfect 1 hertz signal. Well, how about the sample rate? How often are we sampling in this picture? How many times per second do we sample? And you're looking at this, you're saying, okay, 1, 2, 3, 4. It looks like it's either like if it's 5, or actually this, you can see I've kind of color coded this here. It's not 5. The correct answer is 4, because you have the first quarter second, the second quarter second, the third quarter second, and this last quarter second here. So 4 quarters of a second is 4 samples per second. And I color coded here to indicate you have the first second in red. This point right here at 1 second is actually part of the beginning of the second second. And so there's four orange ones there, and then we'd go on and we'd have the third second's worth of data, and that would begin with the yellow going onwards from there. So we have a 4 hertz sample rate. Now, if we have this question, if we want to do this here, what your computer needs to do to sort of interpret this signal is it's going to connect the dots by basically telling you that the signal goes through all these points. And so I have an example here. How well has the computer done if the white signal is what it thinks goes through those dots? Not very well. That's not good. What about now? If I sampled here once every second, so I only had the first one at time 0, and then at 1 second, and then at 2 seconds, if I only had those points, this would be a pretty good fit. But it doesn't fit all 4 of my data points that I have, or all 5 of them that I have on this screen right now. So I need to move it a little over. How am I doing now? Ooh, better. Still not there. That's the signal we wanted, the one that goes through all those points. But now, how many points do you actually need in order to uniquely define that sine wave? Being sure that you know the exact frequency of that wave. So if you only had the 1 point, we showed that you could fit through here 1 point per second. That means you'll have the time 0, and at 1 second you'll have another point. And you can fit that beautifully with this curve that's like the actual curve, the 1 Hertz wave that we're talking about. But you can also fit it equally beautifully and going on into the future with a ½ Hertz wave, something that's slower than that. If you throw in a second point, you get the 1 Hertz wave that goes like the true cycles that we have. You can also fit it here with a different 1 Hertz wave, one that starts on the way down and goes up. So not uniquely defined, but did we get the frequency right? It's actually a 1 Hertz wave, and no matter what, we know that it is a 1 Hertz wave. And so with 2 points, all it takes is 2 points and you can get the frequency right. If you go to 3 points or 4 points, you can get the exact amplitude and perfectly ups or downs right. And so you can define it with 3 points and 4 points, and that helps there. But really, it only takes 2 cycles per second. 2 points per cycle. It only takes 2 points per cycle to define a frequency correctly. And what that means is if you sample up to, say, 100 Hertz, you can detect waves that are up to 50 Hertz, that are 1/2 of that, because you won't be able to detect a 100 Hertz wave because you wouldn't be sure if it was the 100 Hertz wave or a 50 Hertz wave, and you wouldn't be able to tell the difference between those things. So you need 2 points, and this is called the Nyquist frequency, the highest sample, or highest frequency you can detect with digital samples, and it's the Nyquist frequency is half of your sample rate. So if you sample at 100 Hertz, you can determine frequencies reliably up to 50 Hertz. If you sample at 40 kilohertz, you can find bat signals reliably up to 20 kilohertz. And this is really important because we need to decide and tell our microphone how often to record data. And if we want to record bats that are pretty high frequency, and in fact, actually, if I ask this question here, before I get to the bat thing, what happens if there's a signal at a higher frequency? So if the true signal looks like this, we have all these points. Well, I've got my same 5 points here in the first, second, and the next yellow one there, the same 4 points there. How am I doing with this white signal? Does this fit the data perfectly? It does. It goes through every one of those dots perfectly. And so if there's a signal above the Nyquist frequency, your computer is not going to be able to tell that it exists because it can fit that same set of data exactly perfectly with a lower frequency signal. And so you're going to get confused. And so we want to have the Nyquist frequency high because if there's signals that are up high, we don't want to accidentally interpret them as lower frequency signals. So we want it as high as possible. But at the same time, my hard drive fills up really fast. Right now, a day's worth of data, if you collect 24 hours worth of data, it takes 64 gigabytes or so at 384 kilohertz, and that's a lot of data. That's like filling up the hard drive of our computers that we have in our lab every 2 or 3 days. That's not going to work. So we need to come up with a way to tone things down a little bit. So we have to choose our sampling frequency intelligently. So what sample rate do we need in order to reliably record bats at the highest frequency that they make? And so you need to remember, how high up do bat frequencies go? Well, we can see in this little symbol here, we're seeing the harmonic here, which we haven't talked much about, but that goes up to almost 100 kilohertz there. And so if you wanted to detect a 100 kilohertz wave, you'd need to be sampling at least twice that, 200 kilohertz. So you'd need to be sampling 200,000 times a second and recording those numbers and have the microphone sample those at that rate. And so we are looking for something that is, typically we're recording close to 200,000 times a second. In fact, the number is 192,000. But again, if we go higher, we're going to be able to detect both higher frequencies. Sometimes these bats go up higher. In fact, some bats even go up almost as high as 200 kilohertz, which means you need to go up to 400 kilohertz to sample those really high frequency bats. Thankfully, none of those live in our area, and we don't have to worry about going up quite so high. But now that you understand this idea of sampling, we have to go back to our R values, or our R script, and we have to figure out, what do these X values mean? Well, I showed you here that our wave object is sampled at 384,000 times per second. So the amount of time it takes to go from this dot to the next dot is 1/384,000th. And so I can actually tell R that. I can say time between samples equals audio.samp_rate is where that's stored. Oops, and I need to go make that a better. And that's the sample rate is 384,000. I need to do 1/384,000th of that. So that's the time between samples. Well, that's pretty cool. And so what we have to do then is make it X values. And I want to create a complicated sequence. And so to do that, I can take from my handout that I have this way that we can make a sequence from 0 to something by something. Well, we can make that go increasing every time by the time between samples. And so we just need to figure out what's the highest value then. From 0 to that, we need to go to how many samples are there? Well, there's length of y samples times the audio at samp.rate. So this is going to be the number of samples that we have times the, no, I'm wrong. It's actually going to be by the time between samples. So we have this many samples of length of y and the time between them. And so if I hit enter here, let's see how that works. And I'm going to do the length of x and see how I did. 3,842. Uh-oh. So it turns out my x value and my y value are not the same size. So if I try doing plot x comma y, it says, uh-oh, x and y lengths differ. That's not good. How can I fix that? It's because when I made my sequence, I went from 0 to this length of y, and starting at 0, it turns out that what you need to do here is you need to go, oops, we need to subtract off 1 from that. And now when I do my length of x, it's 3,841 data points, and when I plot x comma y, let's see what happens. Ooh, look at that. It shows me the number of seconds now on my x values here from 0 to 0.01. That's a hundredth of a second, or 10 milliseconds. So I have 10 milliseconds, and so now if I do that, type equals L, and xlim equals from 900 to 1200, like I did before. Oh, it doesn't work, of course, because I'm no longer 900 to 1200 seconds. What was I thinking? Let's go back to plotting our xy. What we wanted to actually look at is we wanted to look at from 0.04 seconds, let's say to 0.05 seconds. I want to actually do 0.004. I wasn't looking at my thing carefully. And there we go. Now we're pretty good here. We've gone from 40 milliseconds to 42 milliseconds to 44 milliseconds. And this is really hard for me to keep in my head. So you know what I think I might try and do? I might actually take my x values here, and I might multiply them by 1,000. So x is x times 1,000. That's going to get me from seconds into milliseconds. And now let me plot x comma y and see. Oh, look at that. 10 milliseconds. That's easier for me to work with. So let me go ahead and do that with type equals L. And now I'm going to do my x limits from, let's say, from 4 milliseconds to 5 milliseconds. That's 1 millisecond long. Well, there they are. Excellent. And so now I can count how many wave crests do I have in between there. And I may not be quite having this here, but let's count this first one as 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. It looks to me like I have 20 wave crests that are in here. And so that means that my frequency would be 20 wave crests in 1 millisecond is going to get you 20,000 wave crests in 1 second, or 20,000 hertz, 20,000 kilohertz, or sorry, 20 kilohertz, 20 kilohertz. So we just were able to figure out the frequency of these waves in this example file here just by counting the wave crests. So there's also a wave exploration 2 and 3, and I want you to take a look at those and try and work with those and see if you can count wave crests in those. But sort of after you do, we're going to take a look at how we can get one of these spectrograms, because wouldn't it be easier to figure out the frequency from just asking it to plot the frequency for us? And the way that it does that is it uses something called a Fourier transform. And a Fourier transform converts from that time domain where we have a graph where we're looking at how the signal goes up and down over time, and it converts it into another way that you can look at this signal, which answers a different question. The time domain question is, how much does the energy change over time? Well, there's another reasonable question to ask, which is, how much energy is there in different frequency bands? That's the frequency domain. And what you can see is that this signal here that goes up and down like that is actually composed of 3 different perfect pure sine waves that go have nice, simple, clean cycles to them, and they're all added up together, and you get to here. And so what you can do is you can have a Fourier transform that will look at the signal and kind of do its fancy matrix algebra to figure out what are the signals that go into that. What perfectly pure sine waves can you add up to get this signal? And so you basically do. You zoom in on a small section of the amplitude signal, and you might see something like this. And maybe this one's really clear. Can you see how many waves there are that are perfectly pure sine waves that are added together here? There's one at this low frequency that goes it goes about 1 time every 1.5 seconds or so. Maybe that's every 1 second, actually. Maybe it's once a second, probably it is. And then there's this other one that goes a bunch of times a second. And so those two are added up. And so your Fourier transform will figure that out for you and say, there's 2 signals in here, one at the 1 hertz range and one at the 10 hertz range. It looks like it goes up and down 10 times in that second. And it figures that out. And then what you do is you can then sort of color code those things and figure out the amplitude extremes, like how much energy was there at each one of those. The amplitude of the 1 hertz wave is much higher than the amplitude at the 10 hertz wave, and so you can assign colors to that. And this is, of course, not what this bat signal looks like, but then you would take those colors and you'd put them onto the signal, this graph right here, so that the color represented the amplitude. And then you can move on to the next time window over and do the same thing and the same thing and the same thing, and we'll be able to show how the frequency content of this signal changes over time. And the top figure here is called the oscillogram. It's showing the amplitude over time, and it has a long period where the amplitude's about the same. It's about the same color for a long period in the spectrogram, but the frequency content is shifting throughout that. And so the time domain up on top shows you one piece of information, and the frequency domain at the bottom shows you the other piece of information. So how does R do it? Well, conveniently, if you go to our handy dandy cheat sheet, if you go to our handy dandy cheat sheet, you can see the plot spectrum is here. So let's take that, copy that over into RStudio, paste it in, but we're not quite ready yet because we need to set some values on here. So the plot spectrum is the audio is the name of the audio variable, which we called audio, so we can leave that the same. But the minimum x value is the time, and this is going to be in seconds. So we'll start at time 0, and we have 10 milliseconds. So let's go ahead and type that 10 milliseconds in as the end. That's 0.010. And then what frequency ranges are we interested in for the y value of our spectrogram? We want a frequency, let's look at the frequencies from 0 to 100 kilohertz. And let's see what happens here. The next thing is called the detail level, and that we can play with a little bit, but it's usually a number between 256, 512, 1024, some of these factors, powers of 2, and the last thing is we can set the start. We're going to leave that off right now because it's actually an optional parameter is what that way it looks like. So we can ask it to plot the spectrum like this, and let's see what happens. Woohoo! You can see that there exists this dark black band here representing the highest energy. Right at about, oh, look at that, right at close to 20 kilohertz. And that is exactly what we'd expect for our 20 kilohertz wave. It has that spectrum that's at 20 kilohertz. We can do this with our script. We can just change the name to wave exploration number 2 and hit enter. Oh, it gave us an error, subscript out of bounds. Well, if we look at this, the number of data points we have right here, if we ask it to show us the audio file information, it's only 1,921 samples. Our last one was twice as big, so it looks like we need to go to a shorter time interval. That was my mistake there, is that I'm doing the whole thing, but the whole thing is shorter in this file. So let me go 0.05. Oops, 0.005 is 5 milliseconds. And now, oh, look at that. Now you can see that this signal includes a 20 kilohertz and also something that looks like it's higher than that, like about twice as high. There's this weird kind of red striping in there that is kind of interesting. And what I want to show you is here, if we increase our detail level, that starts to go away here, such that that's not actually real. And now you can see the signal here is showing us those two different frequencies that we have, and that other stuff is arbitrary. You can see I can keep going up by another power of 2, and what happens is that, oh, it says error. It's because I went up too high. I don't have enough high enough sample rate to allow us to go any higher than what I had before. So we can go back down to there. So that's what that detail does. Is we can talk a little bit about the nuances of it a little bit later. But now you've got this. Now let's actually look at a real BAT file, and we'll take a look at it. So I've changed mine to bat1.wave here, and we're taking a look at it. It takes a long time to plot this because there's lots and lots of data points in this file. Remember, it's got 384,000 data points in every second. So it says it's done, but nothing's appeared yet. Wait for it. It'll be here. There we go. So that took quite a while, but you can see each one of these little spikes here is an individual call of a bat. And so here we are, and how many seconds do we have? We have 1.5 seconds. That's because I've correctly converted my x values into seconds here. That's 1.5 seconds. So maybe we should zoom in here a little bit on this so that we can do our plot. And let's change the x limits. And then we have this little C command, means combine these two things here. And so combine, so where do we want to zoom in on? Let's find the biggest, strongest pulse in here. And so that's going to be somewhere around, let's say 0.9 to 1.0 seconds. We'll go ahead and see what happens if we try that. And hopefully it will plot a little bit faster this time. Okay, there's that signal. Now we can zoom in a little bit more. We realize that actually that's going to be from 0.95 to 0.98. There we go. Now we're starting to see. We can zoom in even further, and we can really see the shape of that call. It has a little bit of beginning to it, and some ending in that peak, beautiful sound. And this is the amplitude of this signal. But what about the frequency? Well, to do that, we're going to use our plot spectrum command again, just like we did before. And we were going to use the audio file. We need to tell it the time in seconds. And so let's look at this same little time interval from 0.95 to 0.97 seconds. And the frequency range from 0 to 100. And we'll keep our detail level at 256 to start and see what it looks like. And we do it. And there it is. Look at that. Ooh, look at our signal going all the way up to 100 kilohertz. What happens if we go up to 120? Can we see it? Oh, there we go. Now we can see that you can see a little bit more up there. There is a little glitch that I'll fix later where you can see I asked it to go up to 120 kilohertz, but it didn't change the legend. So stay tuned for that little fixing thing that I'll update that. But let's go back to 100 so that at least we're accurate here. And so we're seeing this signal pretty clearly, which is it's pretty exciting to be able to see the spectrogram in such clear detail. The question is, which bat is this? And so we're going to need to look at our characteristic frequency and other characteristics of this, the high frequency, low frequency. And to do that, you're going to go into Canvas. And in Canvas, under our last week's modules, we have a different file. And that file is called Western North American Bat Acoustic Table 2022. We can open. So the way this file is organized is that it has sort of introduction on the first page, and then it has pages with individual tables. This first table is bats with characteristic frequencies near 50 kilohertz. And then there's one with near 40 kilohertz. Then there's some more bats near 40 kilohertz, 30 kilohertz, and so on and so forth. And so we need to look at our bat and figure out what is its characteristic frequency. We're going to look right before the tail. And this is around in here where it's starting right before the tail. It's going to be around 50 kilohertz is that frequency. So you're going to find the 50 kilohertz table. And you're going to look at these bats. That's the characteristic frequency column. You're going to look at the high frequency column. And so you can look at this and you can sort of scan all the options and say, well, do you think that this is the canyon bat? The canyon bat's highest frequency is usually in around 62 kilohertz. We're up above 100. Now, the canyon bat typically goes, even though 62 is its average, it can go between 51 and 73, and then even below that, they've been observed over the range from 44 to 101. So you can have this happening, but it's less likely to happen with that. So we can start saying, well, it's probably more likely to be one of these other bats, and we're looking for something that has a higher frequency range. Probably one of these two is the highest frequency, and then we can start looking at the low frequency. Well, the low frequency is somewhere around 45, which is pretty close to both of those. Don't get a huge amount of detail and information from there. The frequency at which the maximum energy occurs at, those are, again, very close for those two bats. The duration, well, you have to figure out how long is this signal lasting. It's going from right here, 1 millisecond, 2 milliseconds, 3 milliseconds, 4 milliseconds, 5 milliseconds. Looks like somewhere between 5 and 6 milliseconds. It's a little bit longer, which could happen for this bat, but seems like it's more likely to happen for this bat. And so we've got that there, and we can kind of ignore the slope characteristics right now. Those are things that we're not going to look at as much. And then look at the special characteristics. And so trying to figure out, is there a FM sweep in a smooth curve? That means frequency modulation. Is this a smooth curve? Or is there a more pronounced knee? And that's kind of a relative judgment on that. But that would be one way to tell them apart. And the other thing You can look at all these different details. So you have to decide which bat that you think this is, and you then identify it as either Myotis humanensis or Myotis californicus, which have these 4-letter abbreviations MyU and MyCa. MyU is for Myotis, MY, and U for humanensis, and MyCa, Myotis californicus, the CA. So basically every one of these 4-digit abbreviations is the first 2 letters of the genus and the first 2 letters of the species merged together. So that's how this works. Your assignment is to figure out all of the bats in our 12 bat sequence. They're all listed in Canvas, and you can find them in one zip file that you can unzip for yourself right here at Batwave Simulation Wave Files. So that is one of your homework assignments. Good luck.